tail. So again, lots of genetic diversity. So it's all random.
All right.
All right. Okay. And so this is something that we've kind of already talked about. Again, this is, oh, here's that number. It's 8.3. I lied. It's not 8.2. So 8.3 something million possible ways that these homologous chromosomes could line up. And so this is showing you different alternatives and what those gametes would be as a result. So in the first example, well, it's alternative 1 right here. We have all of dads on top and all of moms on bottom. So then during meiosis 1, you're going to have either all mom's chromosomes or all dad's chromosomes. And then at the end of meiosis, not the end of meiosis 1, but the end of meiosis together. And so alternative 2, you have 2 moms on bottom and 1 of dad's on top. And so your different gametes would be a mix of mom and dad's genetic information. And then if you look at alternative 3, same thing. Now you've got 2 of mom's on bottom and 1 on top. So you get this combination of alleles. And then this alternative is the other alternative. So the way that these homologous chromosomes line up, one pair of homologous chromosomes lines up totally independent of a different pair. And so for each pair of chromosomes, you're going to have 2 ways that it can arrange. Either mom to the left and dad to the right, or vice versa. Or mom to the dad, and then mom to the top, dad to the bottom, or vice versa.
With that slide, we just really need to be mindful that there's so many different variations. We don't have to know, all the different alternatives. We just need to know.
No. It's just used to illustrate a point. Okay. That depending on how those homologous chromosomes line up is going to dictate what type of DNA you have in your gametes at the end of meiosis.
So when you have a child that looks like extremely like you, there wasn't much crossing over?
Not necessarily true, because not all genes are what you look like, so it might be like at those particular regions, and it also depends on your genotype. If you have blonde hair and your husband has blonde hair, you're going to have kids with blonde hair. It's just the way it is. If you have kids with brown hair and brown and brown, that's going to make brown, And so it just depends on the trait, and it doesn't mean that crossing over didn't occur. It just means that, again, depending on the genotypes of you and your husband, and then also depending on whether they do exchange. It's a lot of what ifs. There's no simple answer. Okay, so this is something else that we mentioned. This is, if we said that each gamete is 8.3 million, so forth and so on, genetically unique individuals, when you cross 8.5 million, I forgot to do a math step in here. So if you take 8.5 million and square it to itself, so 8.5 million times 8.5 million, you end up with 70 trillion. So 70 trillion different possibilities of one egg being penetrated by one particular sperm. And so we call this random fertilization, because it's not like the sperm enter a line and it's like, okay, now it's my turn. They're fighting, they're competing. So depending on which sperm penetrates that egg, and all sperm are going to be different, just like all eggs are going to be different, one zygote is technically 1 out of 70 trillion different genetic possibilities. So lots and lots and lots of things can happen here. Okay, so we're going to look at the difference between twins real quick. So I know y'all have heard of identical twins and non-identical twins. So identical twins, what's going to happen, you're going to have one sperm fertilize one egg, and it's going to make one zygote. But then somehow that zygote at some point in the development is going to just split into 2 zygotes. So they're going to have the exact same genetic DNA, because they're still getting the same DNA in that sperm combined with the same DNA of that same egg. So they're going to be genetically identical. So it started off as one zygote and morphed into 2. Whereas the fraternal twins, instead of starting off as one zygote, what happens is there are 2 eggs that accidentally get released during ovulation. And so they're each going to be different, right? Because this is one gamete, this is a totally different gamete. Just like the 2 sperms that are going to penetrate them are going to be different. And so that's why if you have fraternal twins, they might not look exactly the same like identical twins do, because their DNA is going to be different because they've got DNA from 2 different sources. You know, each egg cell is going to be genetically unique, just like each sperm cell is going to be genetically unique. Yeah?
But there's a 1 in 70 trillion chance that fraternal twins could be identical.
Yes. Yes.
Good to know.
I don't think there's 70 trillion individuals on this earth, though. Maybe one day. Okay.
It'd be rather apparent if there was.
It was, yeah. All right, so here we're comparing mitosis to meiosis.
What was the other, the fraternal twins called, the scientific name?
Oh, so, yeah, dizygotic
Okay,
and monozygotic. I didn't mention it because I wasn't going to test you up.
Okay.
So, but, I mean, yes, that's the technical scientific name. 1 zygote or 2 zygotes. Okay. So comparing and contrasting meiosis to mitosis, again, mitosis is going to have 1 cell division, meiosis is going to have 2. Mitosis is going to start off diploid, and it's going to end diploid. Meiosis is going to start off diploid and end up haploid. You're starting with 1 cell in mitosis, and afterwards you end up with 2. Starting with 1 cell in meiosis, and afterwards you end up with 4. In mitosis, these are going to be genetically identical. You're just copying the cell. Whereas in meiosis, we've interjected all of this crossing over and this genetic variation, and so that's going to make all of your gametes, you know, genetically unique or genetically individual, I guess you could say. And so in meiosis 1, we're separating the homologous chromosomes, the mom and dad's chromosomes from one another. And then in meiosis 2, that's where we're separating those sister chromatids from one another in order to make the genetically unique gametes. Do you want me to make a list of differences between mitosis and meiosis? Yes, probably wouldn't hurt. Okay.
That's a good short answer.
Yes. Short answer question, right?
Put a star next to this one.
Yes. All right, so mitosis and meiosis. I'm pretty sure we've already done this once, but it won't hurt to do it again. Let's make a little room. All right. So mitosis, 1 cell division. Meiosis, 2 cell divisions. Mitosis, you're making a clone of a body cell. In meiosis, you're making unique gametes that are not going to be clones of one another. Mitosis, you're going to start off with 1 diploid cell, and you're going to make 2 diploid cells. In meiosis, you're starting off with 1 diploid cell. And then in meiosis 1, you're separating the homologous chromosomes, so you're going to end up with 2 haploid cells. And then those are going to undergo meiosis 2, and you're going to end up with 4 haploid gametes, either sperm or egg. And again, in mitosis, in anaphase, we're separating the sister chromatids from one another. Here, we have 2 anaphases. We've got anaphase 1, and we've got anaphase 2. In anaphase 1, what are we separating? Homologous chromosomes, right? And then meiosis 2, anaphase 2 looks exactly like mitosis. So we're separating the sister chromatids from one another. Yeah, Daniel?
You know, during subdivision in meiosis, after the first meiosis, they're diploids, right?
No, they're going to be haploids after the first division. And the reason is because if these are our homologous chromosomes, you know, crossing over has already occurred in prophase. But then during anaphase, we're going to separate these into two different cells. So this cell is going to have either mom's chromosome 1 or dad's chromosome 1. Not both, because that's what we're separating. And so in order to be diploid, you have to have this complete pair of chromosomes. You have to have both versions, mom and dad's. But these, since we just separated mom and dad from one another, that's why these cells are going to be haploid. Other questions? All right. Okay. So when we're talking about the movement and the arrangement of these chromosomes, lots of different things can go wrong. Most of the time it doesn't, but occasionally something does go wrong. And so sometimes you can end up with extra copies of genes. For instance, the trisomy 21 or Down syndrome, you end up with an extra copy of chromosome 21. So instead of having 46 chromosomes, you've got 47. And so if there was a human that had 3 sets of genetic information, they would be triploid. They would have 69 chromosomes. So 23 plus 23 plus 23. Of course, that doesn't exist because it gets messy. But then also, you can have errors in the way the chromosomes separate from one another. So for instance, in meiosis 1, when you're separating the homologous chromosomes from one another, sometimes these guys are kind of adjoined at the hip. So they're supposed to be separated, but one guy will go over to the other side. And so that means you'll have one gamete that has 2 sets of genetic information, and then another one over here that has no sets, because it's totally missing that homologous chromosome. And so we're going to go through and we're going to talk about different ways that this can happen. And this process is called non-disjunction. So disjunction, if something's at a junction and you're unjunctioning it, you're pulling it apart. But if you're not pulling it apart, then they're staying together. So non-disjunction basically means that the homologous chromosomes aren't being separated from one another. They're supposed to be, but there's an error, and they just don't feel like being separated. And so you can see the results. So here we have a cell undergoing meiosis, and so this is a pair of homologous chromosomes. And so normally in meiosis 1, this guy would come over here, and that guy would go over there. So this non-disjunction, this can occur at anaphase 1. And then if it goes on and makes gametes, you know, this one's going to have a diploid gamete, so that when you make a gamete, or sorry, a diploid egg or sperm, and then after fertilization, you're going to end up with an extra set of DNA, which is going to mess up the individual. And so if you have both of the homologous chromosomes in this, then that means this gamete isn't going to have any of it, right? And so that's going to affect the individual. So they're going to only have one set of genetic information. And so if you can't get these homologous chromosomes to separate from one another, then you're going to end up with an extra set of genetic information in one and a completely missing set in the other. Now, non-disjunction.
What does that cause, like, in humans?
What does it cause? I mean, trisomy 21 is an example. It's an example of non-disjunction. So chromosomes 21 are aligned, and at meiosis 1, they end up separating from one another. And then in anaphase 2, so this non-disjunction, this can occur in anaphase 2 between the homologous chromosomes, or it can happen between the sister chromatids. So when non-disjunction is pretty much when you have 2 pieces of DNA that are supposed to be separating, and they don't separate. They both go to either one cell or the other.
Is it the microtubules that stops it? I mean, are they not strong enough? What stops them from dividing?
I don't know what actually causes it. Maybe the microtubules aren't connected to the centromere well. I really don't know. I mean, that's a good educated guess. I'm sure there's probably lots of different studies been done over, and there's probably lots of different things that can cause or ultimately lead to that. But it's just an error. It's just a mistake. And so, I don't care that you know the exactness of all this, but what I do want you to know that non-disjunction is when you have pieces of DNA, either homologous chromosomes or sister chromatids, that don't separate correctly in anaphase. Whether it's anaphase 1, whether it's anaphase 2. And so this non-disjunction is going to lead to abnormal gametes. Some are going to have extra DNA, and some of them aren't going to have the entire set of DNA that they need. And so this picture right here is looking at the same thing, but instead of non-disjunction occurring in anaphase 1, this is showing non-disjunction in between the sister chromatids. So in this case, so this is meiosis 2, we're supposed to be separating the sister chromatids from one another. But instead of getting 1 here and 1 there, you end up with 2 here and 0 here. So then after fertilization, you bring in your male, and now you've got an extra copy of a chromosome. Whereas this one doesn't have any copies of that chromosome. So it messes up the number of copies of DNA, or copies of particular genes that you're supposed to have. And so again, this can occur either in meiosis 1 or in meiosis 2. So during anaphase 1 or during anaphase 2.
But it's only in the anaphase part?
Yeah, because it's when they actually separate, which they're separating, or supposed to be separating in anaphase. And so an example, like we said, Down syndrome, he's got an extra copy of chromosome 21. And so that's because of this non-disjunction that occurred. There's also some other, I don't want to say necessarily diseases, but conditions that have a third copy of chromosome 18 or a third copy of, chromosome 13. But trisomy 21 or Downs is going to be the most often, the one that happens more often. And then you can also have these alternative chromosomes due to non-disjunction. So you can have males that have 2 Ys, you can have females that have a Y, you can have males that have an extra X, so it gets a little complicated.
What does that mean?
These are they're all various different conditions. They're just genetic diseases, essentially. And so we're not going to get into the differences just because of lack of time, but each one, I think XXX is called Turner syndrome.
Oh, where you
Triple
don't grow and you look like a little girl.
Something like that.
I watch Law and Order.
Law and Order, okay, yeah. So each one's different. All you have to do is Google like XXY. I don't know all the names and what the
filters.
yeah. A lot of them can live and function normally. They're just sterile. They're not going to be able to reproduce because there's just too much stuff going on. It gets confused. It doesn't know which chromosome, anyways, it just gets complicated. Now, there are other types of chromosomal or abnormalities that can occur as well. So you can get this non-disjunction where the DNA is not separated appropriately. You can also have a deletion. So just accidentally, a chunk of your chromosome goes missing or gets cut out. And so you're missing a couple of your genes right here. So if you're missing a couple of genes, that means you're not going to have the DNA to make the RNA, which means you're not going to have the RNA to make the protein. Another example of a chromosomal abnormality is a duplication. So here, if the normal gene should be A, B, or order of genes should be A, B, C, D, E, F, G, here it's A, B, C, D, E, F, D, E, F. So we've duplicated a certain region.
So is that like frame shift?
It's kind of like frame shift, but since you're doing it a gene at a time, each gene is already in its frame. So you're not changing the reading frame. You're literally just like making a copy of a couple of genes and tagging it on. So instead of having one version of the F gene, now you're going to have 2 copies of the F gene. And then you can also have inversion. This is where the order of the genes gets flipped around. So instead of being A, B, C, D, E, F, G, the FED and the DEF have switched places. So in other words, it's A, B, C, F, E, D. So a few of the genes are being turned around in the opposite direction. And so it changes the order of the genes on the chromosomes. So you can have these individual chromosomal abnormalities where you can be missing a piece of DNA, you can duplicate a couple of genes, or you can reverse the order of some of the genes. All right, last slide. So translocation is going to be the other type of chromosomal abnormality. And this is what's going to happen when pieces of DNA that are not homologous chromosomes are switching pieces with one another. So the idea is the same as crossing over because you're just switching pieces of DNA. The difference is in crossing over, it's occurring between homologous chromosomes. In translocation, it's happening between non-homologous chromosomes. So in other words, chromosome 2 from mom and chromosome 7 from dad. So they're switching pieces of DNA, but they're not even the same genes. So that's going to make a mess. So instead of having an individual that has these individual genes, now you end up getting a part of chromosome 2 on chromosome 7, and vice versa. So it messes up the genetics of it a lot. And so you can see they've dyed these different pieces of chromosomes, so you can see that the translocation has actually taken place, but the thing that makes it different from crossing over is that it's occurring between non-homologous chromosomes. So chromosomes that aren't supposed to be paired to one another. All right, questions?